Okay, let's get started. Thank you, everybody, for trying to keep the break on time. And it's exciting to see everybody interacting so much. So I'm going to keep the introduction of our next speaker short because I think we all know Bob. I was going to print out his CV and bring with me this morning, but I felt that it was environmentally unsound to actually print this thing out. Traditional thing. We're happy to have Bob here on the West Coast. Bob's an East Coast guy. Princeton for bachelor's, MD, and PhD from Hopkins, a little time at Harvard, and then back at Hopkins where he's a professor and a Howard Hughes investigator. You know the importance of what Bob has done for our field. The fact that there are latently infected CD4 cells came out of his lab. The fact that they persist despite antiretroviral therapy, he's told us. Mechanisms by which defective and intact viruses interplay. All these are so important for what we'd like to do. So we're thrilled that you accepted our invitation, and we look forward to your talk.
Thanks very much, Keith. Thanks to Keith and Hans-Peter for the invitation to be at this conference. It's really exciting hearing the incredible work that's going on in this field. So I'm going to talk about barriers to HIV cure and how we measure progress towards eliminating them, and I hope that some of the things we've learned in our effort to understand how to measure viral persistence may be useful to you in thinking about gene therapy approaches. I will be mentioning an assay for which these disclosures are relevant. So the best understood barrier to curing HIV infection is a latent reservoir in resting CD4 cells. And we think this is established when, on rare occasions, activated cells become infected just as they're transitioning back to a resting memory state. And in resting cells, key transcription factors, host transcription factors required for HIV gene expression, such as NF-kappa B, NFAT, and PTEF-B, are either absent or sequestered, so that as the cells make this transition from an activated back to a resting state, HIV gene expression is almost automatically extinguished, and you end up with a stably integrated but a transcriptionally silent form of the virus in a long-lived memory T cell. And that allows the virus to persist through the normal mechanisms that maintain immunologic memory, including, as we'll see, proliferation of infected cells. And then, of course, if these cells become activated again in the future, they can begin to produce a virus. Now, there's no sort of clinical assay for this reservoir. The way that we know these cells are present in patients is through a viral outgrowth assay that's really kind of based on this model. So to detect latently infected cells, you reverse latency with a T cell activation. And this is done by purifying resting cells, and these cells don't produce any virus, but you plate them in limiting dilution and then activate them with the mitogen so that cells with the latent provirus can begin to produce virus, and then that spreads through the culture so that in about 2 weeks you can grow out from a single latently infected cell enough virus to measure by a simple ELISA assay, and then you go back and calculate the frequency of latently infected cells. So it turns out these cells are present in everybody, but only at a frequency of around 1 in a million as assessed by this assay. So it's a small population, but longitudinal studies show that this population has a very slow decay rate, a half-life of 44 months, essentially guaranteeing lifetime persistence even in the setting of optimal antiretroviral therapy. Now, these original studies were published in 1999 and 2003, and interestingly, David Margolis has repeated this work recently in patients on newer regimens and come up with essentially exactly the same half-life, which says that all of the remarkable improvements in antiretroviral therapy have not actually impacted this fundamental problem, which is, the persistence of a non-replicating form of the virus. Now, it was clear early on that these cells are widely distributed. We got the same frequencies of infected cells in blood and lymph node, whether we measured total HIV DNA or cells that could produce a replication-competent virus. And interestingly, in both blood and lymph node, the frequencies of cells with HIV DNA were much, much higher than the frequencies of cells from which we could induce replication-competent virus. And I'll come back to that point in a minute. Now, most of our studies have focused on resting CD4 cells, and the reason is that the biology of these cells favors viral persistence, and that's really for two reasons. First of all, as I mentioned, the transcriptional environment in these cells allows the virus to remain latent. And second, resting cells have a long half-life. We found virus inactivated cells, but most activated CD4 cells die during the contraction phase of an immune response, 99%. So essentially, they don't represent a long-term reservoir for the virus. Now, another indication of viral persistence is a trace level of free virus in the plasma of treated patients. This is called residual viremia. It's typically about 1 virus particle per mil of plasma. And we showed that intensification of antiretroviral therapy cannot reduce this residual viremia any further because essentially it's coming from cells that were infected prior to the initiation of therapy. And we did a series of sequencing studies which, as far as I can tell, have never been read by anyone, but we showed that this trace level of free virus in the plasma is actually archival virus that is drug sensitive. It continues to be released for years without any further evolution. And all of this is consistent with the idea that the residual viremia simply represents the daily activation of a small number of latently infected cells. They produce virus which can't infect other cells because of the drugs, but which can be detected with a sensitive enough assay. So measurement of residual viremia actually is one way to assess the viral reservoir. The problem is there's really no dynamic range in this measurement because it's already so low. But the analysis of residual viremia did provide one real surprise. So these are sequences from resting CD4 cells from a single patient on treatment over time. And here are sequences from the residual viremia in the same patient. In some cases, they match, which is consistent with the idea that the residual viremia is coming from activation of latently infected cells. But in this patient, and about half the patient we studied, the real surprise was that the residual viremia was dominated by clonal sequences. That is, identical sequences that appeared without a single nucleotide change in multiple independent limiting dilution amplifications over a 2-year period. And we thought the only way to explain this is that there must have been proliferation of infected cells which would copy the viral genome without error into the progeny cells. And so this work in a similar study by Lisa Frankel's group really provided the first evidence for what we now think is a major barrier to cure, and that is that the very cells we're trying to eliminate actually can proliferate in vivo. And here are just some other examples of these predominant plasma clones, as we called them. So I'm going to come back to this problem of proliferation a little bit later, but first I want to talk about other ways to measure the reservoir. So this viral outgrowth assay, although it always works, it requires 2 to 3 weeks of tissue culture work in a BSL-3 lab, and therefore most investigators have resorted to simpler DNA PCR assays to detect the provirus. Some time ago, Steve Deeks and my wife Janet compared PCR-based assays to the viral outgrowth assay and basically found that the PCR assays give infected cell frequencies that are a lot higher, about 300-fold higher than the viral outgrowth assay. So what does this mean? It means that there's a lot of proviruses that are not induced to produce replication-competent virus in this assay. We call these non-induced proviruses. And of course, the critical question is, do we need to worry about them in terms of cure? Are they inducible? So to address this question, Yachi Ho in our group carried out a full-length single genome analysis of HIV proviruses that were present in tissue culture wells that were actually negative for viral outgrowth. And this was done by a single, at the single molecule level, by a near full-length outer PCR, nested inner PCRs to confirm that we were at limit dilution, and then a series of overlapping PCRs to allow us to get the full sequence. And when you do this, if you have an intact provirus, you get a set of bands that look like this for the inner PCRs, but you also see gels that look like this, where some bands are smaller than expected and some are missing. This would be indicative of a deletion at the 5' end of the genome, and similarly, there are 3' deletions. So when you put all these sequences together, this is what the proviral landscape actually looks like. So in this slide, each horizontal bar is a single provirus, and the white areas are mapped deletions. The green are hypermutated proviruses, and intact proviruses are shown in orange, and you can see that they're actually very rare. So the landscape of proviruses that persist is dominated by highly defective proviruses. And I think you can see that if you simply do PCR, for example, in the GAG region, which is the most commonly used assay, you essentially miss all of these defects that are sort of outside of or encompassed by the region that you amplify. Now, we've also recently analyzed proviral landscapes in treated SIV infection, HIV, and HIV-2 infection. And in each case, the proviral landscape is dominated by defective sequences. There are some differences, for example, compared to HIV-1, SIV and HIV actually show a higher fraction of intact proviruses, and this may have something to do with the time scale of SIV and HIV infections. The deletions for both HIV and SIV tend to be large. On average, half of the genome is deleted in these deleted proviruses. The deletions appear to arise during reverse transcription due to a template switching event. There's actually regions of homology at the deletion junctions. The hypermutation is extensive. It's shown here for SIV. You can see a gradient. There's actually two gradients separated by the central polypurine tract. This is something that Ned Landau had proposed. And what's really remarkable is sort of the extent of hypermutation. So the graph on the right shows the fraction of all of the Gs in the whole genome that are mutated to A. And for some proviruses, it's up to 40%. So this is really sort of a catastrophic level of mutation. So these hypermutated proviruses and these proviruses with large deletions are dead for replication, and we don't want to include them in measurements. Now, I think the most important thing that Yachi discovered, though, in the analysis of these full genome sequences is shown in this slide. And this is, I use Venn diagrams to sort of give a quantitative idea of the frequency of different types of proviruses. So if you use a standard GAG PCR assay, the number of infected cells you detect is something like 1,000 to 10,000 per million. The viral outgrowth assay is typically around 1 per million. And most of this difference is due to these defective proviruses that I mentioned. But Yachi also found that there was a very significant number of intact proviruses that lacked any obvious defects. And this is typically about 100 per million. These had functional LTRs. They were not methylated. They were integrated into genes that were actively expressed in resting and activated CD4 cells. When Yachi painfully reconstructed about 10 of these, they all replicated normally. So nothing to indicate that they could not potentially be induced and replicate in vivo. And to determine whether these intact, non-induced proviruses, as we call them, could be induced, we carried out experiments in which we simply took cells from wells that were negative for viral outgrowth and re-stimulated the cells with mitogens. And even though all of the cells in these cultures proliferate, some of the latently infected cells don't produce virus until after a second or a third or even a fourth round of T-cell activation. And so what it really looks like is this. Each time you activate the cells, you get out additional proviruses, or out additional viruses that become activated. So this gave us the idea that really the best way to measure the reservoir was to measure the total number of intact proviruses, sort of regardless of their transcriptional status at any given point in time. And that's the basis of the new assay that we've recently described. So to develop an assay for intact proviruses, we took information from 431 near full genome sequences, only 2% of which were intact. And we analyzed them sort of gene by gene and found that for most defective proviruses, they're actually defective in most viral genes. And that's because the deletions are large and the hypermutation is extensive. So this is the gene by gene analysis, the fraction of the genes that are non-functional. For protein coding genes, this would be due to deletions, defective start codons, or internal stop codons. And if you consider genes that have to be spliced and defects in the splice sites, the fraction's even higher. So, for example, for TAT, which is critical for high-level viral gene expression, 95% of the proviruses have defective TAT. And this is relevant for cure interventions, particularly if you need to turn on a viral gene expression to get rid of the provirus. Cells with defective and intact genomes are going to actually behave very differently. So we can also ask with this information, what do standard PCR assays tell us? And these are sort of the positions of some amplicons used in standard PCR assays, and we can ask what fraction of the genomes detected by standard PCR assays are actually intact. And for all of them that are in the literature, it's extremely low, less than 10%. And the problem is that if you have an intervention that is selective for the intact proviruses that can actually express viral genes upon activation, you may miss a successful intervention. And this graph shows what percent of the signal would be lost if you had a selective 10-fold reduction in intact proviruses, which would be remarkable if we could achieve it. In fact, the loss of signal would be very low with standard assays. So the problem is standard assays will miss effective interventions. And this is why we need an assay for intact proviruses. So it turns out to be surprisingly easy to do this with digital droplet PCR, where you distribute DNA from infected cells into nanoliter-sized droplets so that each droplet gets, at the most, a single provirus, and then you can interrogate that provirus at multiple informative positions with multiplex PCR. And we found that we could very effectively distinguish intact and deleted proviruses with just two amplicons, one in the packaging signal, which is the frequent site of small deletions, and one in the envelope region, which also contains a conserved area where there are two adjacent ApoBec3G consensus sites that are hypermutated in over 95% of hypermutated proviruses. So we can also distinguish hypermutated proviruses. And turns out there's 13 different patterns of hypermutation in this region, and we developed discrimination probes that would distinguish them with about 95% accuracy. So this assay for intact proviral DNA works like this. There's an optimized DNA extraction, and then droplet formation, and then digital droplet PCR. And the readout looks like a flow plot with fluorescence from the packaging signal amplicon on the y-axis and from the envelope amplicon on the x-axis. And so in the upper left quadrant, you see proviruses that have a deletion at the 3' end, and the lower right is a deletion at the 5' end. The hypermutated proviruses end up in the upper left quadrant due to that's where the hypermutation discrimination is. And the intact proviruses can be directly counted as droplets in the upper right quadrant. And then there's also additional PCR, digital droplet PCR for a cellular gene that allows us to determine the input cell number and also correct for any shearing between the two amplicons. With this assay now, we can get a lot closer to the true number of intact proviruses. It turns out that the assay does detect some defective proviruses that have small deletions between the amplicons, but it's certainly much better than the standard PCR assays shown in yellow. So we've now, and the real advantage is that this assay is scalable. And so we've been able to apply this to large numbers of patient samples, treated patients, and here's the frequency of cells with intact proviruses among resting CD4 cells in a large cohort of patients. It averages about 100 cells per million, which is exactly what we predicted based on the full genome sequencing. And it's a lot lower than the frequency of cells with defects at the 5' or 3' end of the genome. So this is actually the first evidence with a method that doesn't involve a long-distance PCR that most of the viral genomes are defective. There's a pretty good correlation with the viral outgrowth assay, even though the ratio between infected cells detected with this intact proviral DNA assay and the viral outgrowth assay is about a 100 to 1, again, what we expected. And this number is really, I think, critical. It's the small fraction of proviruses that are inducible, even though they appear to be intact, and I'll have a little bit more to say about that. And then we've also been able to look in blood and paired blood and lymph node samples and see essentially similar frequencies. Now, as I mentioned, the real advantage is you can analyze large numbers of patients, and here's a correlation plot for repeat measurements on the same patient about 4 years apart. These are patients on suppressive antiretroviral therapy. So there's a very good correlation between the measurements. Any dot that falls below the line indicates some decay over the 4-year period. And interestingly, a lot of the samples fall on this line, which represents a half-life of 44 months, which we had predicted based on the viral outgrowth assay. But with analysis of larger numbers of patients, you can begin to see outliers. These are people who have very rapid decay and also cases in which the size of the reservoir appears to increase, probably through proliferation. And so we're now trying to understand the clinical correlates of rapid and slow decay of intact proviruses. So we've gotten closer to being able to measure the number of intact proviruses, and I think now comes the hard part. We have to understand what is this difference here. So why are so few of the intact proviruses turning on, even with full T cell activation? As I mentioned, there may be, with repetitive stimulation, you may be able to induce additional proviruses, suggesting that there may be sort of a stochastic element to induction. That is, each provirus has a certain probability of turning on when you activate the T cell. And it's also been suggested by Rafik Sekli and others that some cells may be in sort of a deeper state of latency, for example, central memory cells. And so we've looked at the inducibility of these proviruses in different canonical T cell subsets. We start with purified resting CD4 cells for reasons that will become clear, and then through sort of standard sorting protocols, obtain naive central, transitional, and effector memory cells, and then put them into these long culture experiments in which there's multiple stimulations with the cells being split after each stimulation and half the cells receiving an additional round of T cell activation. And so then from this we can determine how many stimulations are required to induce a given provirus. The starting population analyzed by the IPDA assay shows slightly fewer cells with intact proviruses among naive cells, but roughly similar frequencies among the memory populations. Now, what we had expected was that proviruses in transitional and effector memory cells might be easier to induce, whereas proviruses in central memory cells might require additional stimulations. But when we did the analysis for these 4 populations, we didn't see any pattern that looked like that at all. There was tremendous variation from patient to patient and no consistent relationship between the canonical T cell subsets. And if you look at overall inducibility cells, the number of cells from which you could get a virus out after multiple stimulations, it really wasn't much different among the memory subsets. And if you normalize by the number of intact proviruses, the inducibility perhaps slightly higher for effector memory cells, but differences not really significant. So, and the important thing is that even after multiple stimulations in the effector memory subset, we still have only 10% of these proviruses turning on. So this is a real problem, for example, for a shock and kill type of strategy. Now, one of the problems with this subset analysis is that the markers that are traditionally used to define these subsets actually change with T cell activation. For example, CCR7 goes up dramatically upon T cell activation. And so unless you start with resting CD4 cells, I think the results are very, very difficult to interpret. And this caveat really applies to many of the studies in the literature. It would obviously be better to define these subsets based on the whole transcriptome rather than just a couple of genes. And with single-cell RNA-seq, you can find distinct populations based on the whole transcriptome, but they don't actually correlate with the conventional T-cell subset markers. So we need better ways to analyze this problem. We've also looked at inducibility in blood versus lymph node, and again, don't see much difference. So it doesn't appear that this low inducibility can be explained on the basis of T-cell subsets or anatomic location. What are some other possibilities? Well, one possibility is that there are cryptic defects in some of these apparently intact proviruses that are not obvious from full genome sequencing. That's certainly a possibility. Some of them could be permanently silenced by epigenetic mechanisms. That's also certainly a possibility. And I think while we're sorting this out, I think it's certainly better to measure the total number of intact proviruses than to use standard PCR assays shown in yellow, which are mainly measuring defective proviruses that are irrelevant to cure. Now, I want to point out one additional recent finding that is related to the viruses that come back in treatment interruption studies. There are a couple of recent studies that have compared the rebound virus to viruses detected in the viral outgrowth assay before treatment interruption. And so far, they appear to be different. Now, that could be sampling, but to fully explain this observation, I need to go back to this phenomena of proliferation of infected cells, which was first indicated by analysis of residual viremia. Now, definitive evidence for the proliferation of infected cells actually came from studies done by Thor Wagner here and Frank Maldarelli looking at integration sites. And these studies really prove that infected cells proliferate in vivo. The problem is that integration site analysis is only capturing the ends of the viral genome. So most of the expanded clones detected in these studies are likely to carry defective proviruses. So to determine whether cells with replication-competent proviruses could proliferate, we and others carried out the viral outgrowth assay on large scale and obtained many independent isolates, which we then compared by full genome sequencing. And despite the high degree of viral diversity that was present in these patients, all of whom started treatment during chronic infection, the isolates obtained in the viral outgrowth assay showed a striking degree of clonality. And in this study, 57% of the isolates, independent isolates, had a matching sequence in the same blood sample. And that, if you think about it, that really requires an enormous degree of in vivo proliferation. And John Mellers and Michelle Nussenzweig independently essentially did the same experiment, and all three groups came up with this value in between 50 and 60% of isolates having a matching sequence. So really an extraordinary degree of clonality. So this, interestingly, I think adds to the still ongoing debate about whether antiretroviral therapy completely stops ongoing replication. We think that it does, at least in adherent patients, because the vast majority of sequencing studies show no evolution. Certainly ours show no evolution. Treatment intensification doesn't reduce residual viremia or improve outcomes. Drug resistance in people with access to optimal treatment is actually disappearing. And 3-drug regimens are actually, very surprising to me, but now being replaced to some extent by simpler 2-drug regimens, all an indication of the remarkable efficacy of antiretroviral therapy. And to that, we can now add the fact that the vast majority of cells in the viral reservoir are actually not even generated by direct infection. They're generated by proliferation of previously infected cells. So that changes our view of the reservoir. You know, we used to think of it as essentially a collection of these quiescent cells, but what it actually looks like is this, these very large clones, and probably based on work from Josh Schiffer and Dan Reeves, many smaller clones that are simply not apparent at our level of sampling. And so we really need to understand this process and what drives it. And one of the most, I think, potentially disturbing observations that came from the integration site analysis was that the proliferation may be, was the suggestion that the proliferation may be driven by integration of the virus into certain host genes that are involved in some way in proliferation, and alteration in expression of those genes may lead to continual proliferation of cells. So to look at this, we've analyzed these clones longitudinally using the viral outgrowth assay to obtain replication-competent viruses. And some of the clones persist, as shown in the colors here, which is the fraction of the isolates that represent a single clone in different colors. Some of them persist on a time scale of a couple of years, but others appear and then disappear, so that these clones are sort of waxing and waning on a time scale of months to years. And here's another example. This is looking now at the residual viremia instead of a replication-competent virus. So in this patient, at the first time point, this clone was dominant. A year later, it's replaced by one that's actually ancestral to the first one. And then a year later, another one becomes dominant. So these clones are waxing and waning on a time scale of years, which is really not consistent with the idea that there's some kind of continuous cell autonomous stimulus to proliferation. So underlying the apparent stability of the latent reservoir, there's actually some very complex dynamics with clones proliferating and then clones waning, and the total size of the reservoir remaining roughly constant. And this raises a number of interesting questions. So what stimuli drive the clonal expansion? We don't think that in general it is due to some kind of cell autonomous oncogene-like effect related to the site of integration, but I think a lot more work needs to be done there. Do the stimuli that induce proliferation induce viral gene expression? And is each cell in the reservoir capable of this enormous degree of clonal expansion? God help us if that is the case. And what causes the contractions? How can we accelerate those? So I want to conclude just possibly on a hopeful note. I mentioned that if you analyze the viruses that come back during treatment interruption and compare them to viruses that you grow out from the viral outgrowth assay before treatment interruption, they tend to be different. Now, I don't think that means you can ignore the viruses you would detect in the viral outgrowth assay. They clearly can go, they're essentially one T-cell activation event away from exponential in vitro growth. So I find it hard to believe that we can ignore those. But how do we explain this observation? Well, the simplest explanation is it's just sampling, right? But if a significant fraction of the viruses you obtain in the viral outgrowth assay represent a single clone, then why doesn't that one rebound? And I just want to point you to two interesting recent papers that may provide some sort of a clue. I mean, one idea is that for a clone to get very large, it must not be activated very often. But that's not consistent with the finding of these clonal populations in analysis of the residual viremia. Rather, there may be some control, some ability of the immune system to control these large clones. And the top paper is by my colleague Joel Blankson, who has studied an elite suppressor in whom there is a very, very large clone that is effectively controlled by a strong CD8 positive T-cell response. And then the other paper is from Beatrice Hahn, which suggests that during transmission, type 1 interferon responses essentially sieve viruses during transmission and effectively allow only a subset, those that are interferon resistant, to replicate. So it could be some type of cytolytic T-cell or innate immune response that is actually controlling some of the viruses in the reservoir. If we could figure out how to extend that control to all of the viruses, then we would have a functional cure. So let me just conclude that the vast majority of proviruses persisting in treated patients are defective and therefore accurate evaluation of cure strategies requires assays that are selective for intact proviruses. The reservoir is widely distributed. Interventions that target specific subsets or anatomical locations are, in my opinion, only going to have limited benefit. The low fraction of intact proviruses that are induced by a single round of maximal T-cell activation presents a real challenge for shock and kill strategies. Now, if you can directly target the integrated provirus, as we heard from Tricia, then that problem may not be so great. But the vast majority of the reservoir is generated by, not by direct infection, by proliferation of previously infected cells. And this is something that I think we really have to worry about. I apologize for not having a community summary slide, but I will just say that our assays for measuring the latent virus are getting better. A big problem is that we don't have a good way to turn on the latent virus. And as I said, that may not matter if we have a ways to cut it out with these new genetic technologies. But something that we do have to worry about is that the cells that harbor latent virus can divide and increase in frequency in vivo. So let me just thank the people involved in this work. I've been working on this problem with my wife Janet for many years. Yachi Ho did all of the initial work on defective proviruses, and she's now starting her lab at Yale. Katie Brunner and Greg Laird were involved in the development of the intact proviral DNA assay, and Greg now runs a startup company that's offering a highly validated version of this assay for investigators in the field. And I should say I have no financial relationship with this company. Nina Hasmani and Jennifer Kwan did the work on proliferation, and Alex Bender did the work on SIV. I'd like to thank many collaborators. Steve Deeks has been intimately involved in a lot of this work, as well as Greg Laird. And the work on SIV was done with Dan Baruch, Jeff Lifson, and Louis Picker. And I'd like to thank the funders, particularly the Martin Delaney Collaboratories and the Gates Foundation. And I'll stop there. I'll be happy to try and answer any questions.
Lots of time for questions.
Let me fire one out at you, Bob. The CAR T literature has shown us two examples now with the. Is it on? Hello. So the CAR T literature has shown us two examples. One is Carl June had the TET gene for insertion and expansion of a CAR T population, but then it didn't transform. It quieted back down. And now the NCI group with the CD22 CAR had a Sybil insertion site with their CAR T vector that expanded and was 50% of the peripheral T cells and then shrunk back down. And might what you're detecting here actually be part of the biology of T cell memory? And I'd like to know if you're actually inserting in T cell specific or T cell memory specific regions that expand this pool. And actually, you're kind of detecting the same thing, which is a natural expansion that the vector is showing you, or in this case, the real live virus is showing you, of how a T cell expands and contracts through its natural biology.
Yeah, so Remus, thank you for that question. So my wife actually was the first one to define integration sites in vivo in resting T cells. And it turned out the virus integrates all over the genome, but generally in active transcriptional units, usually in introns of active genes, and most of which are not directly involved in any obvious way in cell proliferation. But more recently, there have been a couple of genes that keep coming up in these integration site studies. And it's clear that I think there are some genes for which an integration event will alter the biology of the cell. But I'm not convinced that that's the general explanation for the proliferation. I think it's more likely that it's sort of memory T-cell homeostasis, where cells expand and contract. And that's something that we're working on right now.
But is it showing you the genes that are important for that memory cell stasis?
No, it's not showing specific genes. Rather what it's showing is particular antigens that drive the proliferation.
Okay.
Maybe two quick questions related to that. One is, do you know whether there's a bias in the reservoir cells that are proliferating toward HIV reactivity?
It's not dramatic.
Okay.
Yeah.
And the second quick question is that do you, those of us who fantasize about antibodies doing something on their own, have to assume that there is some reactivation ongoing regularly from most cells that are latently infected? Are we living in a fantasy world?
You mean to preserve the antibody response to?
And it was for the antibody response to be effective without an LRA. The virus has to show itself
Right.
fairly regularly with enough frequency to have an impact.
Yeah.
Does it?
There I'm not so sure. I mean, whether or not you can use antibodies alone to target the reservoir, without some kind of latency reversal, I'm not convinced. I mean, I think, for example, the fact that these very long rebound, delay until rebound cases suggest that cells can remain in a latent state for years and then reactivate. So my own personal opinion is that you're going to need some kind of way to turn the virus back on. Before the elegant strategy you've developed will be effective.
So, is the clonality of the T cells, when you have this clonal population of the virus, and you go back and try and look at the memory T cells from whence it derives, have the similar clonality when you sequence the TCR? Have you looked at the TCRs?
Yeah, so we're now doing that, and Josh Schiffer and Dan Reeves are helping us to do that. The problem is that with TCR sequence analysis, you get a vast number of TCR sequences, and then you're asking, in the tiny subset of all those T cells that are infected, do you see the same kind of clonality? And how do you do that in a way that sort of accurately scales from a tiny subpopulation to the whole population is something we're trying to work out now. So I guess I said we don't have the answer to that yet.
When you do limiting dilutions, you make it possible to get a defined number of TCRs that would be defined with the clonal population you're looking for.
So
the virus.
One simple way to look at it is with the viral outgrowth assay, we can find infected cell clones that represent 50% of the entire population of infected cells. You never get anywhere near that percentage with, let's say, T cells specific for a particular antigen or T cells with a particular sequence. That suggests that it's
that very early on, it's therefore a single population going into multiple different kinds of T cells and creating
Yeah. I mean, I think that this antigen specificity of cells in the latent reservoir probably, reflects all of the antigens to which somebody's exposed from the time they were infected, with, of course, some being particularly important, let's say CMV, with chronic stimulation and so forth.
So splitting the reservoir, the same reservoir sample in multiple parallel T-cell activation assay, do the viral isolate come up each round show some order? Some viral like come in the first round and some come late, if in the parallel experiment?
Yeah, so we had thought we might be able to see something like that. It turns out if you look at, for example, these clonal populations, all of the cells within a given clone don't behave the same way. They don't all come out after the first stimulation or after the fourth stimulation. So there's no apparent pattern, even though those cells should have the same T-cell receptor and the same HIV integration site. So there's something that's different in the cells that's not determined simply by the sequence of the provirus or where it's integrated or the specificity of the cell. Something more complicated.
Bob, I just wanted to point out one thing. So there's something in the literature that claims that lentivectors and HIV have the same integration patterns, the same frequencies. So going back to the first question about CAR Ts, that's kind of an important caveat to that kind of comment, but let's assume that it's correct. That gives you a good number for the frequency of those kind of events, right? Because there are two examples of this. In patients receiving about 10 to the 8th to 10 to the 9th transduced cells, and maybe approaching 10,000 people treated worldwide in CAR T type examples. This is an extraordinarily infrequent event.
Yeah.
And I think that to presume that could impact this is really not correct.
So you're saying that you don't think that integration site is a major driver of proliferation?
No, it may be found after very long time selection in individuals,
Right.
but in the critical part of the formation of the reservoir, probably not,
Yeah.
I would guess.
Yeah.
But I would also say that there are natural discontinuities in the T cell receptor repertoire, even in a naive immune system, right? And so that determines like individual resistance to certain chronic infections. Like individuals, because of their MHC genetics, are predisposed to be either more resistant or more susceptible to things like herpes viruses. This plays out in many examples, post-transplant care, all that kind of stuff. So that is a highly discontinuous environment, and it doesn't actually require some true antigen. It's the nature of the fact that the TCR has to recognize the MHC at some point in thymic selection, and that creates a discontinuity.
Yeah, sure.
What is the genome copy number in the average latently infected cell?
Well, in general, it's 1. You know, infected cells are rare, so it's in general, most cells don't have more than 1 provirus. John Coffin's actually looked at this pretty carefully through very nice limiting dilution studies, and most of the time, an infected cell only has a single.
Do you think that's because having more copies is deleterious to, let's say, competitive proliferation, and so you eventually will only proliferate if you have low copy number? Possibly.
Yeah, so I don't. That may be going on. The end result or the observation is that the copy number per infected cell is generally 1.
So, Bob, I asked Yachi this in Mexico City, Yachi Ho, and, which is, we hear that there's homeostatic proliferation of the reservoir. We hear that there may be these integration site pro-proliferative events, and then obviously there's antigen-driven expansion. In what I'm not hearing, and you've come as close as I think we've heard, is how do you rank these, or how much do you really think is important? And it sounds to me like you really think that antigen-driven proliferation is probably the major driver of these expansions and contractions. Am I sort of paraphrasing your thought?
I think that's right. We're trying to do those studies now. We've certainly seen dramatic expansions of T cells specific for particular antigens that have a clonal provirus present. But to do that with replication-competent viruses, which is only a tiny fraction of the total, it's more difficult. But my sense is that it's antigen. Homeostatic proliferation, the mathematical models are suggesting, it can give rise sort of just almost randomly to large clones, but not of the size that we see. But that's certainly also a contributor to persistence, I think.
And this contraction that we've seen, then, is that something we can manipulate or take advantage of? Obviously you'd love to, but
that's the hope, because the clones, as you grow out in the viral outgrowth assay, that may dominate at a particular time point can be gone a year later, or not. Well, they may not be totally gone, but they're below the limit of our sampling. So indicating a dramatic relative contraction relative to the other ones that are dominated at that time point. So it would be great to take advantage of that, or to block the proliferation, as I think some people here are thinking about. So.
Right, yeah. And there's a question right over here.
So just to expand on that, because if it is antigen-driven proliferation, you are going to have, you should be able to calculate, and maybe you got this from your modeling, what that decay is, because you'll have that expansion and then you'll have that contraction. Whereas if it's homeostatic, you can mostly model what that is just based on some of the earlier studies from Rafi Ahmed, et cetera. So,
Yeah, Yeah, well, I mean, so Rafi Ahmed's work in mice, really gave us this idea that the degree of expansion, and this was mainly for CD8 cells, but could be, such that you go up to, half of all cells recognizing, LCMV or something like that. So I think we need to take advantage of a lot of the work in sort of basic mouse T-cell immunology and try and understand the dynamics of it. But it's a complicated problem. Maybe, I don't know if Josh wants to say anything about this, but he's been thinking about that a lot. And,
I don't have anything definitive, but one of the other problems is discriminating formation of the reservoir versus maintenance of the reservoir. And we're starting to think that those are two completely different processes. One of the things that came out of Rafi Ahmed's lab and other labs is that individual clones undergoing homeostatic proliferation don't do so evenly. And so it's not, I had a misinformed opinion going into this that, antigen-driven would give you this sort of rank abundance where certain clones are proliferating much more avidly than others. But the same thing can happen with homeostatic proliferation for different reasons. So I think to tease it out really does require looking individually at each clone and ascertaining based on TCR or other factors if there is antigen specificity.
Any other questions? All right, help me thank Bob.
thank you. Thanks.